Hello students, I am Ms. Diksha Gaonkar, Assistant Professor in Botany, Government College of Arts, Science and Commerce, Kepe. Program Bachelor of Science, 3rd year, Subject Botany, Semester 5, Course Code BOC 106, Course Title Cell Biology and Plant Biochemistry. Title of the unit Biomolecules. Module name Lipids, Classification and Structure of Lipids and Fatty Acids. Outline Lipids and Fatty Acids, Classification of Lipids and Fatty Acids, Structure of Lipids and Fatty Acids. Learning outcomes. At the end of the module, you will be able to define lipids and fatty acids, classification of lipids and fatty acids, and draw the structure of lipids and fatty acids. Coming to the introduction, so let us see what are lipids. So lipids comes from the Greek word which means lipos is a fat. So these are the heterogeneous group of compounds related to fatty acids and include fats, oils, waxes, etc. The term lipid was for the first time given by German biochemist Blur in year 1943. Fatty acids. So fatty acids are long chain organic acids having 4 to 30 carbons atoms, and they have single carboxyl group and a long non-polar hydrocarbon tail, which gives most of the lipids their hydrophobic and oily or greasy nature. Fatty acids do not occur in the free state or uncombined state in the cells. So fatty acids which occur in the natural fats are usually monocarboxylic. Mono refers to one. And contain a number of carbons atom, as these are synthesized from 2 carbon units. And they are straight chain derivatives. So here you can see the carbon chain we have of 2 types, saturated fatty acids and non-saturated fatty acids. So all the saturated will have only the single bond and the unsaturated will be having more than 1 double bonds. So this is the structure representing saturated and unsaturated fatty acids. So you can see in this structure of saturated fatty acid, there are only the single bonds everywhere present. And this is the carboxyl group that is present and long chain of carbons. Okay, so for every fatty acid, it could be whether saturated or unsaturated, this chain is going to vary. The number of carbons, the number of hydrogen, but this carboxyl group is going to remain same for all the fatty acids. Then we have unsaturated fatty acids. So the presence of the double bond, one or more. There could be 1 double bond, there could be 2 double bonds, there could be 3 double bonds. So the number of double bond also varies, and depending upon that we have different types of unsaturated fatty acids as well. So these are some of the examples of saturated and unsaturated fatty acid. Let us just quickly list them. So we have caprylic acid with 8 carbon, capric acid with 10 carbons, lauric acid with 12 carbons, myristic acid with 14 carbons, palmitic acid with 16 carbons, stearic acid with 18 carbons. Under unsaturated fatty acid, we have maristolic acid with 14 carbons, palmitolic acid with 16 carbons, sapinic acid with 16 carbon, oleic acid with 18 carbon, linoleic acid with 18 carbon, arachidonic acid with 20 carbons. So these are some of the examples to cite under saturated and unsaturated fatty acids. Now let us quickly see the difference between lipids and fats. So lipids, they are the broader group of biomolecules found in the body of an organism, whereas the fats are the type of lipids. They are the type of lipids. Examples under lipids necessary for healthy body. So lipids are, it could be solid also, it could be liquid also, whereas all the fats, they are solids only. Then lipids are the organic compound that forms a framework of the structure and the function of the living cell. Whereas the fats, they are the energy reserves of the body. Then either the simple or the complex, these are the types of the lipids, whereas the fats could be saturated or they could be unsaturated. Then See, the Blur, it's a name of a biochemist. In year 1943, he did classification of lipids. And he classified lipids into 3 categories. We have simple lipids, we have compound lipids, and we have derived lipids. And in today's module, we are going to discuss about all these things. So, under simple, we have fats and oils, we have waxes. Under compound, we have phospholipids and glycolipids. And under derived phospholipids, we have steroids, we have terpenes, we have carotenoids. So, let us start. First one, simple lipids. Simple lipids sometimes also referred to as homolipids. So, simple lipids are the esters of fatty acid with various alcohols. So, fats and oils, so under this first one, we have triglyceride or triacyl glycerol. So, these are the esters of fatty acid with the glycerol or glycerine. And they are the main energy storing compounds in the body. And triglyceride could be simple triglyceride or it could be mixed triglyceride. So, when the fatty acid here we have triglyceride, if the fatty acids of all the 3 fatty acids are identical, it is simple. And if all the 3 fatty acids are not identical, then it will be the mixed triglyceride. So, here we have the structure of triglyceride and monoglyceride. So, this is a glycerol molecule as you can see here, and to that the 3 fatty acids are attached, which makes it triglyceride. Now again in this example you see, this is an example of or the structure of monoglyceride. This is a glycerol and to that only 1 fatty acid is attached. So it makes it mono. Mono refers to 1, here tri it refers to 3. So depending upon like here if 3 are attached, so it becomes tri. Here only 1 fatty acid is attached to the glycerol molecule. It becomes monoglycerol. This is a structure of triglyceride and monoglyceride. Then let us see the difference between plant fat and animal fat. Animal fats, they are saturated. That means they will not have any bond, double bond. Here only single bond will be present. Then in case of plant fats, they are unsaturated. Double bonds will be present. Solid at room temperature. Plant fats, they are liquid at room temperature. Then they are stored in the liver and the bone marrow. These are mainly stored in the seeds and the fruits. Oxidative rancidity is more here. Here it is less. Examples to cite under butter fat, beef fat. Here under this plant fats we have coconut oil, we have castor oil. Second one is waxes. So waxes are the esters of fatty acid with high molecular weight. Okay, and these are monohydroxy alcohols. Highly insoluble in water and chemically inert, and they serve as water barrier in insects, in birds and in furred animals. Compound lipids, they are also referred to as heterolipids. So, compound lipids are the esters of fatty acid with alcohol and they possess additional functional group. So, under these, the first one that we have is a phospholipid. They are also referred to as phosphatides. Now, let us see what are these. The compounds containing fatty acid, glycerol and phosphoric acid, then nitrogen base and other substituent. So, this is a structure. So, the glycerol backbone, as you can see here, the glycerol backbone. These are the fatty acid 1, fatty acid 2, and these are the phosphor containing group. So, this is a phospholipid. Example, phosphatidylcholine is an example here. Now, phospholipid in turn are of 3 types. We have phosphoglycerides, we have phosphoinositides, then we have phosphosphingosides. Okay, these are the 3 types. Coming to the second one, we have the glycolipids. So, the substance whose molecules consist of lipid covalently attached with the simple or the compound sugar residues. So, they are important constituents of brain where their amount is 8% of the solid matter, and other than the brain, they could be present in other tissues as well. So, we have covered simple lipids, we have covered compound lipids, and now we are going to discuss about derived lipids. So, under derived lipids, we have 3 types: steroids, terpenes and carotenoids. So these are the different types of steroids: C29, C28, C24, C19 steroids. Under terpenes, we have monoterpene, sesquiterpene, diterpene, triterpene, tetraterpene, polyterpene. And under carotenoids, we have lycopene, carotenes, and xanthophylls. Coming to the first one here, steroids. So they are insoluble in water. They are soluble in many organic solvents. And they have 4-ring structure, as you can see here, number 1, number 2, number 3 and number 4. 4-ring structure, and they are biologically important because many hormones are coming under this category. So in humans, the steroids, they are synthesized from the cholesterol. Second one is terpene. So among the non-saponifiable lipids found in the plants are the hydrocarbons, and they are the terpenes. And these hydrocarbons and their oxygenated derivatives have less than 40 carbon atoms. And simplest form of terpenes are monoterpenes. There are others also, sesquiterpene, diterpene, triterpene, etc. But the simplest is monoterpene. And we have carotenoids. These are widely distributed in plant and animal kingdom, and these occurs in unsaponifiable residue. This is important, of the plants and animal lipids, and they are isoprene derivatives of high degree of unsaturation. Summary: in this module, we have learnt about lipids and fatty acids, classification, that is simple, compound and derived lipids, structure of fatty acids, that is saturated and unsaturated, structure of lipids, that is a triglyceride, monoglyceride, steroids, and also phospholipids, and difference between lipids and fats, and also plant and animal fats. These are some of the references that I have used. Thank you, students.